Hello, my name is Mohit, and in this video, I'll provide an overview of our paper titled Extracting Causal Visual Features for Limited Label Classification. We thank the technical program chairs, area session, and session chairs for holding this conference in a hybrid format. The link to the paper on archive, as well as a link to our website, which has several other related works, is also here. In this paper, we consider visual explanations in images and show how they have both causal and context features. As the title suggests, we extract and present the causal features. Let's start by defining visual explanations. Essentially, explanations justify some decisions. And if these justifications are visual features, then we have visual explanations. Consider this example. Given the image of the bird and ask the question what bird species the bird belongs to, the answer is it is a spoonbill. The explanation here has to answer why spoonbill. The provided image with the overlaid heat map on the right is the visual explanation. The provided explanation uses a well-known technique called Grad-CAM. It's used to understand why a neural network specifically answered why spoonbill. Let's dig deeper into this explanation. The highlighted red region is why the decision of spoonbill is made. In fact, the red region is more impactful for the decision of a spoonbill than the yellow and green regions. The blue region plays no role in the decision or a very limited one. Consider the language-based explanation in the center. All the necessary characteristics, including the flattened beak, the pale and pink body, and everything else is highlighted in the image. This is exactly what we expect. Hence, the network is making the right choice based on the right features. For this explanation, we used a VGG16 architecture. The framework is given here. The network provides a decision of spoonbill and an explanation of why spoonbill. Let's replace the VGG16 with an AlexNet architecture. AlexNet also correctly classifies the image as spoonbill. However, the visual explanation now changes. This is not uncommon. The change in layers, kernels, architectures, it causes AlexNet to look at different regions to make the same decision. Let's analyze this a bit more. Again, AlexNet has made the right decision that the bird is a spoonbill. The features that it has based this decision are pale pink body, legs, and shallow water. I want to separate between these two sets of features, between the shallow water and pale pink body, which is provided in different colors. Let's compare between the two architectures. The difference seems to be that AlexNet uses the shallow water in its decision process. This feature, shallow water, is an important characteristic of the context where we might find the bird. In other words, if we can take the spoonbill out of the water, then it would still remain a spoonbill. So this reason of shallow water is contextually relevant, but is not the true reason for why the bird is a spoonbill. We call these kind of features as context features. The pink body, flat beak, and straight neck, however, are why the bird is a spoonbill. Note that existence of context features is not unimportant. Presence of shallow water makes the network confident that the object is not a chair, for instance. It can sometimes provide an easy way for networks to contrast between classes. Hence, both the causal and context features are used to make decisions. This is the premise of the paper, that there exist both causal and context features in networks. Our goal, simply put, is to take existing explanation techniques and divide the features provided into causal and context features. Now, I want to answer the question of why we are doing something like this. The first and simple answer is that networks can be compared. It provides a way to evaluate networks from the prediction, and hence accuracy is the same. Human beings are more trustful of networks that make decisions on regions that are causal. The second reason deals with safety critical applications. Consider the CT scan provided here. The shown CT scan is from a patient with COVID-19. A neural network is trained on a data set of these scans. During testing, the given image is presented to the network. Network correctly predicts that the scan is from a COVID positive patient. Let us say that we obtain Grad-CAM results. The overlaid Grad-CAM heat map is shown here. Let us use the Grad-CAM as a mask. We overlay the Grad-CAM heat map on top of the image and only extract the highlighted red and yellow region. If we now pass the masked image through the network, it still predicts the correct class, COVID. Notice the probability, which is 0.79. This number describes how confident the network is that the image is COVID positive when looking at this region. Now, we do the same masking using our method, which we'll describe next. Using the method, we obtain a heat map of causal regions. The masked causal image is passed through the network. The network still predicts that the image is COVID positive, but with a probability of 0.91. Note that our method is not a technique in the conventional sense. We take Grad-CAM and extract causal features from them. The shown results are essentially the causal regions within Grad-CAM. These two heat maps and their associated probabilities can now be used by medical practitioners to see if the explanations matches their expectations and if the network is making its original decision based on causal features. Let's put this in our summary slide. about why the causal feature extraction is required. Let's now discuss about how this can be accomplished. For this, we need to define causal and context features. Once the definitions are clear, we'll provide an overview of the theory and extraction technique. Firstly, both causal and context features lead to some decision P. For instance, the causal features of the pink body, flat beak lead to decision of spoonbill, while the shallow water also leads to decision of spoonbill. Specifically, causal features increases the likelihood of presence of P, while context features indirectly increase the likelihood of P by decreasing the probability of existence of another class Q. Hence, causal features cause P, while context features reduce the likelihood of Q, thereby increasing the likelihood of P. We can actually call these context features as contrastive features. This is because essentially context features provide contrast between other features. We provide some contrastive features here. We can think of contrastive features as explanations that answer specific questions like why spoonbill rather than flamingo. We can see that the contrast features are present at the neck of the bird. Flamingos have an S-shaped neck. Hence, the contrast features show the absence of S-shaped neck, making sure that the bird is now not a flamingo, thereby increasing the probability that the bird is a spoonbill. As a small aside, these kind of contrastive explanations can be obtained by making a small change in the Grad-CAM framework. While Grad-CAM backpropagates logits of predicted class, we backpropagate a loss between predicted class and a contrast class. This was a paper during last year's ICIP from us. You can read the paper or watch the video for further details. The QR code and links are actually provided here. These contrastive explanations exist for other classes as well. We show two more here, crane and a pig. A crane is different from a spoonbill based on the color of its feathers. A pig, on the other hand, is different in its body, legs, neck, beak, and every other part. The contrast features are showing that. Similarly, we can obtain Y spoonbill rather than all learned classes and any combination of these classes. In fact, there are possibly 2 to the power of n such features where n is the learned class. This is essentially the power set of all possible classes. For a binary setting, we need to define 2 to the power of n contexts. In other words, to satisfy our definition of what context is, we need to define 2 to the power of n contexts. Hence, our problem definition is appropriate for a limited label setting, hence the title of our paper. Let's consider a binary case where P and Q are the 2 classes. The possible combinations are actually shown here. Let's understand what these combinations actually mean. PQ are those features that can lead to both P and Q decisions. So for instance, the pink body leads to both flamingos and spoonbills. Shallow water also leads to both flamingos and spoonbills. P bar Q are those features that lead to only decision Q and not P. This can be the S-shaped neck in the flamingo, since it's present in Q, which is a flamingo, but not in P, which may be a spoonbill. PQ bar are those features that exist in spoonbills, like a flat beak, but not in a flamingo, Q. P bar and Q bar are features like white feathers that are not present in any class. With this setup, we describe the theory behind our approach. Let's use CP and BP as the causal and context features. We already know that Grad-CAM shows both the causal and context features. Our goal is to only extract the causal regions, basically the red regions within the Grad-CAM. To do so, we need to model BP, which is the context features. We approximate this based on the power set of contrastive features that we discussed earlier. Notice the P and Q as they vary subscript within the contrastive explanation C. We'll essentially use them as contrast labels. BP is approximated as shown in the diagram. We'll first describe each of the 3 Venn diagrams on the right-hand side. The first one is all sets of possible features in the image. These are features that lead to both decisions P and Q from earlier. The second one is features that lead to neither of the 2 decisions. We can consider them as auxiliary context features that are not important for increasing the probability of either P or Q. The last is features that are causal but are resolved incorrectly. For now, we can think that the network does not classify P with 100% accuracy, which is practical. Hence, P-bar provides features that are the reasons the network is not 100% sure about its decision. Hence, contrast feature is P, a loss between predicted confidence P and Y not 100% P. Therefore, these are the problems in the causal resolution affecting all features. Going back to our goal, we obtain BP by combining these 3 sets of contrastive features. From here, we can extract the causal features. We already discussed that contrastive explanations are derived by backpropagating losses within Grad-CAM framework. For each type of features, we show what is being backpropagated. We pass the image through the network. The final 2x1 logits are presented to a binary cross entropy loss with the shown labels and backpropagated. For PQ, we backpropagate 1, 1. For PP bar, we backpropagate 1, 0. And for P bar, Q bar, we backpropagate 0, 0. For further details, please stop by our poster session. Regarding evaluation, it's not straightforward to evaluate causality and explanations. We provide 3 evaluation criteria here. The first 2 are quantitative and the last one is qualitative. For the quantitative, we use the image masking and recognition strategies that were described earlier. For evaluation, we use COVID CT scan dataset. The dataset is a binary classification dataset with 349 COVID positive and 463 COVID negative scans. We use multiple networks, including ResNets 18, 3450, and DenseNets 125 and 169 in this study. The first evaluation metric, causality, measures causality within a given network. Given an image, the Grad-CAM explanation is derived and the image is progressively masked based on a threshold. This is passed through the network and its accuracy is derived. This is termed as deletion metric. Parts of the image are masked or deleted. The y-axis in the given figure is accuracy. The x-axis is a ratio of Huffman coding between the masked image versus original image. Higher the ratio, less is the masked portion. Hence, the proposed causal features in red has a higher accuracy at a lower ratio. That is, the network is able to identify the class correctly, even when an image with a large masked portion is provided. This is because the non-masked region are in fact causal and is more essential for classification. Ideally, the curves are towards the lower end of the x-axis. The Huffman coding is used since, from an image perspective, eliminating or masking structures is more detrimental to human interpretations, since we are more tuned with structures rather than pixels, and Huffman coding is a proxy for this. For further details, please feel free to stop by our poster session. The second evaluation metric evaluates inter-network causality. We obtain causal features from one network and mask images based on these before passing the masked image through a second network. This measures how well the causal features in one network transfer to other networks. In table 1, we construct masked Grad-CAM images using ResNet 18, and those masked images on ResNet 3450, DenseNets 121, 169 are evaluated. The causal features are extracted, images are masked, and the same experiment is carried out. As can be seen, the accuracy of causal features from Grad-CAM extracted using our proposed method exceeds the Grad-CAM results themselves, thereby evaluating the efficacy of our method. In table 2, we show results when the base network is ResNet 34. The same phenomena occurs here as well. These 2 evaluation metrics show the importance of causal features for a network's primary task, which is recognition. We also show qualitative results for what each of the Venn diagram features from before. Specifically, we show for the universal feature set PQ and the causal resolution set P bar P, and the proposed causal features from the original CT scan image. Note that the explanations themselves are normalized before that being displayed here. Till now, we have shown why causal features are important. Here we show qualitatively the value of context features. In the shown image, both the masked images from Grad-CAM and the proposed causal map are recognized correctly with a confidence of 0.9. However, the context features seem to add value in the sense that they make the explanations human interpretable, which is seen in the overall Grad-CAM image. This phenomena requires further study in a larger setting, both on medical and natural images. Finally, the contributions of this paper are given here. Our first contribution was in the premise of the paper, that current explanations are a combination of causal and context features. We then provided definitions of causal and context features and provided a methodology to extract causal from context features. We also established evaluation criteria, 2 of which are objective metrics. Finally, we demonstrated the importance of context features. While causal features are required for recognition, context features may be important for human interpretability. Please stop by our poster session during the conference. I'm looking forward to meet everyone there. In the meantime, if you are interested in this work, then please check out some of our other works on our website. Also, you can read the paper on arXiv. Thank you.